Welcome to Discover ASAP, the video interview series where we highlight the top findings and tools connected to the ASAP network. Today I am talking with Ricardo Marquez Gomez, a postdoc on Team Cragg, about a microfluidic platform they developed to better study corticostriatal neuron connectivity. So, Ricardo, to start, could you briefly tell us about your overall ASAP project and the impact you hope to have on Parkinson's disease?
Hi, Madeline. Yeah, of course. Thank you for the space. Um, yeah, the overall laser project that we're working with is to study neuronal circuit in human models. We use human-induced pluripotent stem cells or iPSCs to do this. And we also want to apply as many techniques from animal research into in our circuitry. So we're focusing our study in the circuit formed between the cortex, the striatum, and dopamine neurons, which are has been the focus of circuitry research for Parkinson's disease. And we're also quite interested in studying the axons and synaptic physiology and genetics of dopaminergic neurons, which we think is a crucial part of understanding the onset of Parkinson's disease. Now, to do all of this, we have generated or developed a technique called fluid walls, which allows to pretty much treat the circuit as a brain. And when I say that, it means we want to compartmentalize the neurons, so keep them in specific regions, and then have these brain regions talking to each other in an ordered manner as it happens in a brain. Something else this technology kind of, or we hope that we allow us to do, is to have as many patient lines as possible in our research. When studying iPSCs, there's a lot of variability between patient lines. So we hope that this technology will open the door to explore as many patient lines as possible, making the studies more reproducible. And so, yeah, in a way, kind of bringing the high throughput skills into studying circuitry PD research with human neurons.
That's very cool to hear about. Could you give us a little bit more of a brief overview of how this microfluidic platform works, what it looks like, how you developed it?
Uh, yeah, sure. So the technology we work with is called fluid wall microfluidics, and this is pretty much using, instead of solid or rigid walls to generate microfluidic devices. That's usually people use either PDMS molds or commercially available plastic microfluidic devices. We use oil to generate those structures, and the oil we use is called FC40, and FC40 has the property that it can get pinned down to a surface that has some lipid in it. So it creates a water oil interface where you can put cells and grow them and differentiate them. So what we're doing with this circuit is, or with this technology, sorry, is creating circuits in which we have a small chamber that we call dumbbells. Those dumbbells are about 4 to 5 microliters. So we have one chamber connected by a conduit, and then we have a second chamber. All of it is made from oil. And as I mentioned before, one of the features that we're trying to recapitulate with this technology is having this brain-like structure. So for that, we need to first keep the neurons in a specific place, and then just allow them to project axons and synapses in unidirectionally. So for this, the properties of the system allow us to play with the fluidics and the volumes in each one of the chambers. So for instance, when we try to compartmentalize the neurons in one side, we play with the fluidics, so the flow goes against the neurons, and then we can change the direction of the fluidic microsystem to then get the neurons to, or facilitate the neurons to send projections through the conduit.
Wow, that's very cool. A little bit over my understanding, to be completely honest. But do you answered this a little bit? But is there anything that you want to speak about regarding any advantages that this kind of structure might have over existing tools like it? So you mentioned a little bit about like the rigid plastic walls that might be commercially available. Is there specific benefits to doing things this way?
Yeah, sure. So when we started working, I mean, just a bit of background how the thing came into the game. We started working with PDMS molds. That was how we started exploring this corticostriatal dopamine circuitry, um, which kind of worked pretty well. That we get neurons compartmentalized, the same projections to the conduits. It was very laborious and quite time consuming. And when we wanted to ask more relevant questions, more complicated things about neuronal communication, then the system was just not there. So we thought we really needed a technology that allow us to have access to the whole system and that can preserve the connections between the neurons. So that's when we came up with the fluid was microfluidic. That was a team of engineers working in this technology, using it for a very different purpose. They were using it to compartmentalize individual cells and do some cell cloning. But we thought the properties of that technology was quite applicable to generate neuronal circuits. So we started a collaboration, and we've had the first one. There's a publication now that you can see using cortical neurons and shuttle neurons. This beautifully compartmentalizes the axons. In terms of advantages, I think there are three that I would like to emphasize. The first one is the system is not rigid. Again, I mentioned it before, but just want to mention it again, which pretty much means that you can have access to every part of the circuitry. You can access the neurons, you can access the axons, you can access the synapses, and because they're fluidic properties, you can also compartmentalize them very well. The second one is a very adaptable system because the walls are not rigid. That means that you can pretty much draw anything that you like over the oil, and you can also destroy what you draw and then rebuild it. And I think a very good example of this is in the paper. When we put the cortical neurons, we allow them to send projections to the conduit, and then we destroy the system, create the axotomy protocol to cut the axons in half, and then rebuild the system, so I'll allow the neurons to recover from the axotomy. So that's something that at the moment I think no other technology allows you to do. So play around with the circuit as you would like. And the other one is that is a very scalable system, which means that you can pretty much put these circuits anywhere you like. It can fit different experimental platforms. You can, or we can print in 96 wood plates, 35 millimeter dishes, 65 millimeter dishes, MEA plates. So I wouldn't say that one fits all, but I think it has a very wide range of activities. So that pretty much allows you to put the circuits in different platforms, to ask different questions while preserving the whole structure. And yeah, those three.
Yeah, that sounds really interesting, especially seeing the regeneration of the axons you mentioned, which kind of leads me to my next question, which is, are there specific ways that you envision researchers using this platform, or specific questions that you can think of that people might use to address a better understanding of Parkinson's using this platform?
I think that the so the compartmentalization, having the neuronal bodies, the axons and the synapses, and having the chance of exploring particularly the axons and the synapses of dopamine neurons is a very good feature. Um, but I think something that is even more important is the scalability of the platform. Um, I think that in order to understand really what's going on in Parkinson's disease, you have to accommodate as many patients as possible, as many genetical backgrounds as possible, uh, which I think most of the technologies that I know till now are really laborious to achieve that. I think this technology can really help in pushing forward the research of as many patients as possible, and then focusing on exploring the axons and the synapses. Because I think until now it's quite consensus that Parkinson's disease starts by degeneration of the dopaminergic axons. Some animal models are there that can help us to understand, like the mitopark model that's is doing a very good job. In the human side, I think we're quite far away from it yet. So I think this platform might be able to open that door, allow us to have access to the axons and synapses, while at the same time accommodating in a single platform as many patient lines as possible to address the variability issue within of this of this model.
Yeah, it sounds like a major strength here is that being able to look into an actual human axon and kind of just see it in a dish in a way that we haven't been able to really do in the past yet. So that's super exciting, and I hope to see it expand
Yeah,
out.
I hope to see it too.
Yeah. So can you share any tips or tricks for using this platform, or are there special steps of the protocol that require special attention or anything like that?
I'll say that I think I can summarize all of that in maybe time. I think time is a trick. Um, and time in two senses. The first one is working with iPSC neurons means that each one of the subtypes, each, cortical neurons, striatal neurons, or dopamine iPSC neurons, they have different differentiation times. So you want to get to a point where your neurons are mature enough that they are committed to become neurons, but young enough that they are resilient to be placed in this oil water interface, which is it's not the natural habitat, right? So it's quite it can be a bit rough. So just finding the right moment to plate each one of the individual neurons is an adventure on itself. So just getting that right. The second one is time in terms of how long do you need to keep the neurons within the microenvironments until they get mature enough so you can start asking physiological relevant questions. And iPSC research, most models, they push the neurons until 100 days in culture or 200 days in culture. So which means you really need the neurons to be happy for that long. And in order to keep them happy, you need to have a very good substrate so they can attach to. So the coating of the plates um, then becomes super important. And we found that a mix of a very low concentration of PLO mixed with some bio coating, just Geltrex or biolamin or something like that, works pretty well. And the neurons are quite happy living in those conditions. And so far, we've managed to push the neurons until day 17 culture, which I think, at least for dopamine neurons, they're quite mature. We got some calcium signals from them so far, so the happy time. So yeah, my advice would be timing in putting the neurons in the microfluidic systems, and then the coating will affect for how long would you keep the neurons in culture.
Okay, sounds good. Is there anything else that you would like to share that I didn't ask about?
Um, I think I just want to point out the relevance of the interdisciplinary collaboration that we set up to start this project. And just want to give a shout out to the engineers that have done an amazing job in kind of fitting our demands and all the requirements of how the brain works and all the amazing conversations that we had in the exchange of fluidic and neurons kind of environment. I think it's very important that we open the door from different disciplines to join neuroscience and also allow PhD students to explore different options and different ways of making new technology and answering their questions. Um, I think that's the only thing I would like to add on top of everything that we've discussed so far.
That's great. I really appreciate that. It was great to hear about the work that you and your team are doing to further Parkinson's research. And we will put a link to your paper in the notes for this recording. So thank you so much for chatting with us. And we look forward to hearing more from you soon.
Yeah, no worries. Thank you.